Hello everyone. In this lecture, we will be discussing about the biomolecule lipids. We will also look into its classification. We will understand what fatty acids are, and we will understand the different classes of lipids, that is the simple, compound, and derived lipids. And then at last, we will learn about the functions of lipids. So let us start. First, let us see that what exactly is the definition of lipids. So lipids, they are the organic compounds containing hydrogen, carbon, and oxygen atoms. And these are the framework for the structure and function of the living cells. So the lipids, they are insoluble in aqueous solutions, that is water, but they are soluble in the non-polar solvents such as carbon tetrachloride. So if I have to explain this, you might have seen that whenever you try to mix oil in water, it is not possible. That is because of its insoluble property. Whereas if you try to dissolve oil in any non-polar solvent, it will be easily soluble. Unlike the proteins and other biomolecules, the lipids are not polymers. So if you see the carbohydrates, they are the polymers of the simple monosaccharide units. If you look into the proteins, they are the polymers of amino acids. Whereas if we talk about the biomolecule lipids, they are not polymer, but rather they are the small molecules. Now, before we understand the different classes of the lipids, let's see what a fatty acid is, because this is a term that we are going to see a lot when we talk about lipids. So a fatty acid is one of the major components of triglyceride, which is a form of lipid, and we will be discussing about this in the coming slides. Now, the fatty acid is a long hydrocarbon chain with a carboxylic acid end. So what is a fatty acid? A fatty acid is a hydrocarbon chain with a carboxylic end. As you can see here, that this is a long hydrocarbon chain and there's this acidic group present here. And we can also represent a fatty acid by RCOOH, where this R group is standing for the aliphatic chain, and this is the carboxylic group. Therefore, this whole molecule becomes acidic in nature. Now, let us talk about the different classes of lipids. So there are three classes. First is simple lipids, then we have compound lipids, and we have the derived lipids. Simple lipids are basically the esters of fatty acids. As we have just seen that what exactly a fatty acid looks like. So if we make esters of those fatty acids, those are the simple lipids. And under the simple lipids category, we have fats and waxes. Now, what is the difference between fats and waxes? In case of fats, the esters are formed between the fatty acids and glycerol. Whereas when we talk about waxes, these are the esters of long chain fatty acids and long chain alcohols. So this is the major difference. So the difference is between the length of the fatty acids and also the alcohols. So there is this simple alcohol glycerol present in the fats, whereas we have the long chain alcohols in case of the waxes. We will see the structures in the coming slides. The second category is compound lipids. When we talk about compound lipids, these are the esters of fatty acids and alcohol, which contains other groups also. So in simple lipids, there was only the fatty acids and the alcohol. But when we talk about the compound lipids, we have another groups other than these two present as well. Derived lipids, third category. So this category is composed of hydrocarbon rings and a long hydrocarbon side chain. So these are derived from the simple and the compound lipids. Now, there are certain examples under simple lipids. We just saw that there are fats and oils and waxes. When we talk about the compound lipids, we have phospholipids as one of the important examples. We have glycolipids and we have sulfolipids. Under phospholipids, we have lecithin, cephalin, plasmalogen, we have sphingomyelin. When we talk about glycolipids, we have cerebrosides and gangliosides. Talking about the derived lipids, these are having one important component, that is sterols. So all the steroid hormones, they are derived lipids. We have isoprenoids as well, such as the terpenoids and carotenoids, which are the plant pigments. Now let us look into the each category in detail. So the first category was the simple lipids. So talking about the fats and oils. We saw that the simple lipids are the esters of fatty acids with alcohol. And what are fatty acids? These are the long hydrocarbon chain with a carboxylic acid end. So when this kind of fatty acid makes an ester with the alcohol, that becomes a simple lipid. So one of the important examples is triacylglycerol. Now, what does that mean? Triacylglycerol is basically the ester of fatty acid with the alcohol glycerol. So when the ester is being formed between fatty acid and glycerol, then that becomes triacylglycerol. Okay, so talking about some of the different examples of fatty acids that can be present in these fats and oils and any kind of lipid, wherever we will talk about the fatty acids. So these have some examples. So they have been categorized as saturated fatty acids and unsaturated fatty acids. Saturated fatty acids are the ones where only single bonds are present, whereas the unsaturated fatty acids are the ones where there are double or triple bonds present, right? So there are not only the single bonds, we have the double and the triple bonds here. So this is not correct. These are the double and the triple bonds. So some of the examples of the saturated fatty acids include palmitic acid, which has 16 carbon atoms and there are 0 double bonds. So now what does this denote? The one thing written in the bracket. So the first number before the colon indicates the number of carbon atoms in that fatty acid, and the number corresponding to the second digit after the colon, it is the number of double bonds present, double or triple bonds. Then we have another example, stearic acid. So this has 18 carbons with 0 double bonds or triple bonds. So this is also a saturated fatty acid. In terms of unsaturated fatty acids, we have examples such as oleic acid, linoleic acid, and linolenic acid. We have other examples written here on the screen as well. Talking about this next category under the simple lipids, that is the waxes. And we saw that waxes are basically the esters of long chain fatty acids with long chain alcohols. Therefore, the molecular weight of such waxes is quite high. So it's the same thing here. The oils, the fats, and the waxes, they are all the same. The only difference is that in case of the oils, the alcohol was short, also the fatty acids were short in length, whereas in waxes, both of these components, they are longer in the length. And you can see here, this is the fatty acid section, this is the alcohol section, and this is the ester formation between both of them. Now, the natural waxes, they're generally composed of long chain fatty acids esterified to a long chain alcohol that we just discussed. Waxes, they are completely insoluble in water because of the long hydrocarbon tails and their hydrophobic character. And waxes are solid at room temperature. That is why they are solid, because of their high molecular weight. The most important example of the simple lipids is the triglyceride, and this is the storage form of fatty acids inside the body also. So the triglyceride is a lipid molecule which is made up of 1 unit of glycerol and 3 fatty acids. So how do we do that? So this is a glycerol alcohol. As you can see, there are 3 carbons, 1, 2, and 3. 3 of them are having their individual alcoholic groups. Now, when we attach 3 fatty acids to this, it becomes a triglyceride. As you can see, these are the fatty acids. We just discussed that it is a long hydrocarbon chain with a carboxylic acid end. So this is the first fatty acid, second fatty acid, and third fatty acid. These 3 fatty acids, they correspondingly go and do the condensation process and remove water molecule, and there is a formation of bonds. So this is what is formed at the end, triglyceride. So this is the formation of triglyceride. Now, the function of triglycerides is that they are the high efficiency storage fluid fuels inside the body. Now, the triglycerides, they are responsible for storing 6 times more energy as compared to glycogen. So you know that the glucose is stored inside the body in the form of glycogen. But if we talk about the triglyceride, it's holding 6 times more energy as compared to the glycogen. The short chain unsaturated triglycerides, they are liquid at room temperature, and the long chain saturated triglycerides are solid at room temperature. The next category is the compound lipids. Now, what are compound lipids? Compound lipids are the lipids in which, other than the fatty acids and the alcohol, there are additional groups present. And the important examples include number one is phospholipids. Now, phospholipids, they are the primary component of the cell membrane of the cells, right? So now what is the difference between the simple lipids and phospholipids? So as you can see here, everything is same here. There are three carbons. This is glycerol backbone. We have two fatty acids attached here, but instead of the third fatty acid on the third carbon, there is a different group present, and this is the phosphorus group, as you can see here. So because of this phosphorus group present here, this is known as a phospholipid. So other than the fatty acids and the glycerol alcohol, there is a phosphorus present as well. That is why it's called phospholipids. Now, the phospholipids, they have a hydrophilic head and the hydrophobic tail. So the hydrophilic head is basically corresponded to the phosphate group that is present here, and the the hydrophilic head is the phosphate group, and the hydrophilic tails, they correspond to these long fatty acid chains. The next example is glycolipids. Now, glycolipids are the molecules where the fatty acids are having the specific alcohol, stingosine, and a carbohydrate group present as well. That is why they are called as glycolipids. Glyco is referring to the presence of carbohydrates. So when there is a conjugation of carbohydrate and the lipids, then that becomes glycolipids. Now, what is the function of this glycolipid? So glycolipids, they are found on the outer surface of the cellular membrane, where it plays a structural role to maintain the membrane stability. So the phospholipids that we just discussed, they are the major constituents of the lipid cell membrane. Now, on this lipid cell membrane, on the outer surface, there are certain sugar residues present, and these sugar residues are in the form of glycolipids, and they are responsible for maintaining the membrane stability. Also, these glycolipids, they are responsible for facilitating the cell-cell communication because they act as receptors, or you can say anchors for the proteins. So they are important for communication between the cells. There are other compound lipid examples as well, such as lipoproteins. These are the molecules that are responsible for transportation of the fat molecules. So you might have heard about low density lipoprotein, very low density lipoprotein, then we have high density lipoprotein, and so on. All these are the examples of lipoproteins, which are compound lipids. The third category is the derived lipids. Now, derived lipids are the compounds that are produced by the hydrolysis of simple and complex lipids. And the most important example that comes under this category is steroids. They are a type of hormone which are usually recognized by their tetracyclic skeleton. And this is formed by three fused six membrane and one five membrane ring. As you can see here, these are the six membrane rings, and this is a not membrane, member. So there are three six-member chains, and there is this one five-member chain. So total of four rings, they attach to each other, and this is a steroid skeleton. And onto this, when different groups are arranged in different fashion, they become different steroids. So triglycerides, LDL, HDL, VLDL, they are all different types of cholesterol which are found in the blood cells. And cholesterol is an important derived lipid because it is derived from this steroid skeleton only. Now, what is the function of cholesterol in the body? Basically, it is responsible for providing rigidity to the cell membrane. So, for example, let's say these are the phospholipid molecules, which are the major constituents of the cell membrane. The cholesterol gets stuck here and there between these two adjacent phospholipid molecules, and hence provides rigidity to the cell membrane. Here on the screen, you can see the structure of the cholesterol, which is having a total of 29 carbons. Now, what are the different importance for the lipids? So lipids, they are a storage form of energy for long-term use, that is in the form of triglycerides. They have hormonal roles as well, such as the steroids that we just discussed. And the steroid examples include estrogen and testosterone. They are also responsible for the insulation because they form a fat layer around the organs, and they provide cushion kind of thing. And The insulation is both thermal and electrical. So in case of thermal insulation, triglycerides are there. In case of electrical insulation, we have sphingolipids present in the cells. For protection of the internal organs, like a cushion, and it's because of the triglycerides and the vaccines, and they're also acting as the structural components of the cells, for example phospholipids and cholesterol. So that was it about this lecture. Thank you so much.